Hi, folks. Welcome to an introductory to brain function. Sorry, I look homeless, but my office is really cold. Today, we're going to discuss some very broad questions of the function of the human brain and some of the foibles and some of the interesting things. I'm going to share the screen. Switch to that. Share. And go to full screen. All right. So I didn't really know what to call this talk, so I just called it brain stuff. And there's some broad questions that might, obviously that's my name. I'm Kurt Andrus, and I have a PhD in evolutionary neuroscience. And I'm working on another one in the studies of the human cortex. So what does the brain do anyway? And that's not a trivial question. What's the brain made up of? That one's pretty easy. What's the language of the brain? If you think about the concept of an action potential in the brain, it's binary. How do you take a binary signal and get color? It's a picture of my daughter. How do we know that that shirt is blue based on a binary signal in the brain? That's not trivial. How does it talk to other parts of the brain? That's complicated as well, but we'll get to that. How does it develop complexity? Concept of neuroplasticity or changes in the brain is an emerging field. And how do we figure out what it does? And we'll talk about that a little bit. There's a very famous quote down here: if the brain was simple enough to understand it, we would be too simple to understand it. Now, press pause on the recording for a second and contemplate that. It's kind of an interesting question. What's the ultimate limiting factor to understanding the brain? Hit pause and think about that one for a second. And I'll give you a hint. What are we using to try to understand the brain? We've got a degree of freedom problem here, and it's a very difficult one to get our heads wrapped around, no pun intended. Okay, so just what is this brain thing, and how do we figure out what it does? We have imaging. This is one of my favorite MRIs of all time. But this is relatively new. This is within the last 40 years. CT scans were about 40 years ago. MRIs really hit its head about 25 years ago. And before then, how did we figure this out? And it's kind of complicated. So let me start with a question, and I hope this works. It works really well on my large monitor, and it often works with PowerPoint projectors. So hopefully this will work on your computer. You really need to line yourself up and look straight at it. And I've done this a few times with other classes, and I always ask the question, what is this? And people stare and stare at it. And inevitably, somebody always says the same thing, which just fascinates me. It looks like a skull. And I've never seen a skull, but I'm biased because I know what it is. And after a while of folks staring at it, and if you want to press pause and stare at it for a few more minutes, that's fabulous. I'm not used to recording these. I'm used to doing it in person. And eventually somebody will notice, that looks kind of like an ear. That looks kind of like an ear. And there's a couple eyes. You kind of see a snout coming down here, and there's a couple nostrils. Oh my goodness, it's a cow. Yes, it is. Why did we struggle seeing that? We all know what cows look like, but we don't live in a dichromatic world. In other words, we don't live in black and white. And so without any corresponding information around us, it's tough to discern what we're looking at. And once you recognize the cow, then it's pretty obvious. I did the same thing a couple years ago. I took a picture of a Dalmatian and I put it in Photoshop and inverted it. So it was a black dog with white spots and nobody could recognize it, because that's not what we're used to looking at. We have learned that when you see a white dog with black spots and usually blue eyes, that's a Dalmatian. You flip everything, and suddenly our world changes. It's the concept of perception and reception. So how does the brain do that? And we're not really sure. And you're going to hear me say that several times today. So what does the brain do? Well, the first thing I'm going to tell you, we're going to discuss a little bit about how we sense our environment. The brain means very little to us. I'm sorry, our world means very little to us if we can't sense it. The only output of the brain is the motor system. The only input to the brain is the sensory system. And sometimes that sensory system is external, the environment. Sometimes it's internal. My stomach hurts. And we're going to talk about those today in a little bit of detail. That's a dog. I've always been fascinated with dogs just because you just wonder what's going on. What are they sensing? All right. Let's start with some few odd things. This is one of my heroes, Jerry Andrus. I regret that I never had a chance to meet this character. He was a fascinating human being. There's this great quote, "I can fool you because you're a human. You have a wonderful human mind that works no different from my human mind. Usually when we're fooled, the mind hasn't made a mistake. It's come to the wrong conclusion for the right reason." I'm going to show you a quick video. Just watch for a second. All right. I've shown this video to a bunch of folks, and I found that it usually takes the second time around to really catch on. Now pay close attention. All right. Now, normally I would ask the students what, at the very beginning, what does that look like? And inevitably people will say, it looks like a box. And then when you saw him crawl out of it, you realized that it's not a box. It's not even really much of a three-dimensional structure. You notice that board on the back actually goes in front of the board on the front. And this board in the back goes in front of the board on the front and then attaches to the board on the back, and that rope is not attached to anything. So the human mind insists upon putting things into a context that makes sense to us. And that comes again down to the concept of reception versus perception. And is perception necessarily reality? So what is the brain for? This is one of my favorite case studies from a chicken called Mike. And the story is way back, and you can see this. You can go to MikeTheHeadlessChicken.com and read all about this. This is where I took this from. And in the mid-40s, they decided to butcher a chicken for dinner. So they cut the head off the chicken, and the chicken ran around like a chicken with its head cut off. And then something amazing happened. He didn't die, and he kept running around. And the owner of the chicken, Lloyd, thought, well, this is kind of wild. And he ended up taking taking Mike around to county fairs for a while and showed him around, and, you know, for a nickel or a dime or what. Actually, it just says that right there. Lined up to pay 25 cents to see Mike. And Mike lived for months and months and months. He eventually died because he got out of the coop one morning and went out in the rain and drowned. And so I tell my graduate students there's a really inherent important message here, that the function of the brain is primarily to prevent drowning. But it raises, and there's a picture of Mike's head right there, and it's not really attached to the body. 18 months without a head. That raises a very, very interesting question. Do we need a brain? Mike could digest, poop, you know, have normal blood flow, processing, all these sort of things that were under zero control of the brain. So what does the brain actually do? That's kind of a complicated question. And we're going to dabble a little bit in that today. We're not going to answer it all, but we're going to dabble in that a bit. All right. So let's think about performance and movement. I have a ballerina, and then we have Hank Aaron. And I want you to spend a little bit of time thinking for a moment about the sensory structures that are involved right at the initiation of movement, whether it's a pirouette or swinging a baseball bat. And if you want to press pause and think about this for a second, I want you to answer a question for me. Let's go to baseball for a second. How many muscles in Hank Aaron's body are not activated right now? Right? The answer is none of them are not activated. Everywhere from his posture to his legs to his arms. And that raises an interesting question. Then what defines movement? You know, we've taught for years and years and years that movement is defined by muscle activation. Well, every muscle is activated. So how do we create movement? We have to turn off muscles that we don't want activated, which allows those muscles that we do want activated to contract further and control that movement. Now let's switch over to the sensory side. What senses is Hank Aaron getting into him right now? It may be crowd noise, it may be smell, it's certainly sight, but at the same time, we have to have a sensation of body posture and where the body is, and then we have to make a decision. We have to decide how and when and to where to swing that baseball bat. And a 100-mile-an-hour fastball from home or from pitcher's mound to home plate takes 0.6 of a second, 600 milliseconds. So we have to activate all of that in the brain in 600 milliseconds. But we have to maintain motor control so we can move the arms, but at the same time, don't fall over if we miss it. And we're going to talk a little bit about that today. Okay, so this is kind of my theory of how this whole system works. I think I might have stole this concept from somebody else, or maybe I invented it. I don't know. So I give credit where credit is due, but I don't know where the credit goes to. So this is my initial idea of neural control. We have a plan. We're going to decide to swing that baseball bat. Once we make that decision, we activate the appropriate neurons in the human cortex, and that's going to send a signal down to what we refer to as pattern-generating neurons. Those pattern-generating neurons can act independently to create a behavioral output. So let's go back and think of Mike for a second. Mike was not thinking about activating the heart. He was not thinking about walking. He was not thinking about digesting. He was not thinking about pooping. because the pattern generating neurons can work independently, but they get turned on by the plan. But when we create a plan, that plan gets sent over to a part of the brain that I refer to as the state of the body, the condition. So let's go back to an example. Let's say we're going to swing a baseball bat. We come up with a plan. We send that plan down to the pattern generating neurons. We send that plan also over to the state. As the pattern generating neurons are creating a behavior, we're also generating a tremendous amount of sensory feedback. And that's going to go back to the state and say, did we do what we had hoped to do or planned to do? And if not, we have to fix it. So I was looking for a stupid example, and I found a picture of Nazis slipping on the ice. So the plan was to walk across the ice. The plan didn't work right. So the state suddenly changed, and now we have to regenerate this. And I know you guys have all experienced this. You know, you think you're at the bottom of a set of steps, and you go, oops, I missed one step. Suddenly the state changed, and suddenly the plan had to change. That, in a nutshell, is sensory motor coordination, the integration of sensory with motor output. Okay, so go get yourself a blank sheet of paper, or you can just do it in your mind. That's okay. When I go to the next slide, I want you to count all the white dots you can find. Ready? Now remember, count the white dots. You need to be facing your computer monitor straight on. Now, normally this was where I would ask, how many white dots are there? And somebody will do the math and say, whatever that is, 5 times 7, 35. And then I'll ask the students, raise your hand if you see any black dots. And half the class raised their hand. And I said, well, both of those answers can't be right. What is the correct answer? The correct answer lies in perception. So if I asked you to stare at that dot right there, you would see a very clear white dot. But if you focus very intently on that white dot, in your peripheral vision, you see dots over here are becoming grayer and blacker. But then if we switch down here to go to that one turns white. So what's the reality? The reality of it is your perception. And it's a very simple physiological phenomenon that there's a concept in the brain called lateral inhibition. So if we focus directly on something, in order to have high resolution, we laterally inhibit all the information from around us. That's exactly how the retinal ganglion cells in the eyes work. And so we're shutting this off and making everything else look like its background until we focus on that one, and that one turns white again. So it's a very simple phenomenon that can be explained neurologically. And here's another example of perception versus reality. I stole this image somewhere online, I don't remember where. But I want you to focus on a card. Pick a single card and remember it. Any of those 6 cards. And don't forget what that card is, because I'm going to reach through the power of Zoom and I'm going to make that card you picked disappear. Usually when we're fooled, the mind hasn't made a mistake. It's come to the wrong conclusion for the right reason. So your card disappeared. And you know why? Go back and look at this. Now pick 2 cards. King of Clubs and the Jack of Clubs. Neither of them are there. Of course, because none of these cards are over here. But I told you that I was going to make one go away. I made all of them go away. So that's an interesting concept in perception, that we have to define order in our world. Okay, last one. This is one of my favorites. Perception versus reception. So I want you to look at the blocks A and B. And focus on those two blocks. And then what I did, I took Photoshop or PowerPoint, and I blocked everything out except A and B. They're exactly the same. I did this a couple of years ago, and one student in class got so mad at me that I literally had to cut this slide out and put these squares next to one another just to convince her. Let's do that again. They're exactly the same. So, but we perceive them different, even though the reality is they're exactly the same. Why? The same thing with the white dots. Sorry, I'm about to cough. B is surrounded by dark squares. A is surrounded by light color. So by comparison, they're very different colors, when in fact, they're exactly the same. It's a very simple optical illusion, but our perception is different from our appearing. Okay, this one may or may not work. This might take you guys a little bit of a practice. I want you to stare at that chair on the right. Just stare at that chair. And in your peripheral vision, I want you to make that ballerina move in a different direction. Ooh, she's slowing down now. Now she's going backwards. This might take a little bit of practice. You might have to rewind and look at this one a little bit more. Because it's how we perceive movement in the brain too, which is also kind of tricky. Now, let's pay attention a little bit closer to the chair. Which direction is that chair facing? Is it facing this direction? Or is it facing that direction? It doesn't work quite as well on a computer. There we go. Now it goes backwards. There's the back going that direction, and now here's the back going that direction. Okay. So I was given a talk a couple years ago to a group of older folks, retired folks, who were still very, very curious and fascinated with things. And I wanted to talk about curiosity and creativity in science and brain function and things like that. And I thought I would find a picture of clouds. Because nowadays, children don't spend as much time staring at clouds as I think we should. We should all spend time laying in the grass and staring at clouds and seeing images, and anybody with a lick of common sense recognizes that immediately as the embryo of a chicken. But we see shapes in clouds, and we make order out of things. We have to put stuff into a context. We have to make boxes, like Jerry Andrus. And I wrote this: the mind doesn't always interpret correctly, and perception is an individual construct. And I didn't do this on purpose, but right in the middle of the talk of this slide, I started laughing because I made the perfect point unintentionally. Go look at the word correctly. Got it? That was my point. I didn't do that on purpose. But that's exactly the point. I accidentally put in 3 R's, but you didn't read them as 3 R's. You read it as correctly. And since I did that a couple years ago, I've never changed this slide because it's the perfect model for what I was talking about. So, if much of reality is based on perception, let's go to the health professional program for a second. How do you treat a patient's neurological issues if their perception differs from yours? Now, you walk into any clinic and you'll see those little smiley faces, and they'll ask you the question, so on a scale of 1 to 10, how do you feel? Well, I don't know anybody else's perception. Maybe my pain level I perceive as a 10, and the next person would perceive it as a 3. Those are not trivial problems to deal with. So how do we know? Well, let's first start by going to some brain stuff. Excuse me, here's a human brain. It's got all this wrinkly stuff on the outside. And let's dive a little bit into the brain itself. I'm going to add next to it a frog brain. There's this area called the telencephalon, which is all this stuff up here. The optic tectum, which is actually buried inside the human brain. You can't see it on the outside. Little cerebellum here on the frog. There's the big cerebellum here on the human. And the brainstem right here, which is also right there. What's interesting is the brainstem of a frog is not much different from the brainstem of a human. Kind of humbling, isn't it? What's different is the telencephalon, all this big stuff. So why do humans have big brains? It's an interesting question. It also raises the question, why are humans born functionally helpless? Well, it's because of a big brain. We have a giant head that has to get out. And because of that, it's not fully developed. And it takes a while to develop motor skills and sensory cognition and things like that. But this also comes back to Mike, the headless chicken. Here's what Mike had intact. The brainstem and the medulla. The brainstem and the medulla. Those control heart rate, digestion, feeding. We've already talked about all that sort of stuff. Next slide. There we go. So what's the brain made up of? Nobody really knows. Well, actually, the what is pretty simple. Lipids, which are fats, some proteins, lots of water. But when it gets down to the cell types, there's an estimate about 100 billion neurons and about 100 to 500 billion glial cells. Glial cells are an incredibly important component of the brain. And it regulates, it has a metabolic structure, it has a physical structure to regulate the movement of the neurons and the position of the neurons. It also takes up neurotransmitters and sends them back to the other neurons. I'm going to guess in the period of our lives that we're just going to combine glial cells and neurons into one word and just call them all neurons because it really functions the same. But there's only about 100 billion neurons in the brain. And there's only two types. Projection neurons, which go somewhere. So you've got a motor neuron that's going out to a muscle. You've got local circuit neurons that communicate with each other. And in between, to communicate with each other, there's a concept of a synapse. And we're going to come back to that in the next part of this. And there's been estimated that there's 1 quadrillion synapses in the human brain. That's about 10 to the 15th synapses. But let's put this in context. This is equal to about 500 million synapses per cubic millimeter. That's a pretty intense number. So now we've introduced the concept of perception and reception and what's the brain made up of. And now for part 2 of this discussion, we're going to discuss how do we figure out what it does and where it is. So I'm going to stop here and we'll come back next time and we'll continue on how do we figure out what it does.